Yeah. All right, well, I'll try, if that's still too loud. Is that too loud? Okay. All right. So, of course, the big one was Deepwater Horizon in 2010. So this doesn't happen every day, right? And we're still waiting for the Puget Sound oil spill, right? And that's one of the main reasons we're working on this, is because we still live in an area that's at risk for a major spill, you know. So, you know, the spill that happens in the Strait or Puget Sound, these are all to be announced, right? They haven't happened yet. Hopefully they won't happen, but we want to be prepared if they do happen. That's one of the main things that we're trying to do, is develop the tools to really understand what the impacts are. And the biggest lessons from these spills is that we don't really have the tools to understand what both the short-term and the long-term losses are, which for NOAA as a government agency, that's really important, because we're the ones that deal with the responsible parties, and when there is damage, we're the ones that got to get the money back to put into restoration. So to be able to quantify those losses is really super important in order to get money from a responsible party and to put it into restoration. But do oil spills happen every day? They do. This is a little cell phone video that I grabbed. This is a parking lot at the gas station near the Montlake Lab, and you see that little oil spill running right down to the storm drain here. Of course, that storm drain dumps right into Lake Washington, right? So it's that everyday little trickle of oil that we're concerned about. Now, this is back in 2005, right? This was the PI when it was still around, when Department of Ecology, right, with their studies on the pH compounds, which I'm going to talk about in a second. Puget Sound is chronically known for decades to be a hotspot for fossil fuel pollutants. Some of that was, you know, historical, industrial, but ecology was able to kind of track sources and whatnot, and pretty much motor vehicles. So 2005, you know, 12 years ago, I don't think this has changed, right? And if anything, with population increases, it's just getting worse, right? So that's kind of the issue. So our main focus around here, so oil spills are these episodic big events that happen all the time, but as Lyle mentioned, it's the stormwater runoff that for our, you know, everyday things that we're studying, that's one of the most important. So of course, you know, before there was a bunch of us around here, and it was just the tribes, we had this mostly forest, and the forest is a giant sponge, right? So the rain gets soaked up into the sponge, and there's, you know, that's how the ecosystem worked. But then as soon as we started building cities and paving over, what happened is the sponge goes away, we make impervious surfaces, and the way the cities were engineered is almost all those pavement, all those storm drains, they don't go into a sewage treatment plant, they go straight, they get dumped into the nearest stream, and from the nearest stream, it gets dumped into the sound. So the source control is basically all about this. And that's what we're trying to do, is understand what are these chronic impacts of runoff, primarily related to motor vehicles and you know, heavily trafficked roads. How can they be minimized? And our efforts that we're doing to minimize them, are they effective? So really trying to do effectiveness monitoring for this. And we're trying to, from a mechanistic, you know, hardcore biology standpoint, is we're trying to develop diagnostic tools to make these assessments in the field. You know, it's also, so that stormwater runoff, but that's also related to the sewage plant issue, right? Because the way things are designed in most of our cities is there is a little bit of you know, connectedness between the sewage treatment systems and the stormwater system, so that when there's really, really heavy rains, the sewage treatment plants back up and you get these what we call combined sewer overflow, which you can see physically. This is the Montlake Cut, right? So this is going into Lake Washington right near the Montlake Lab. And we've got when it rains really heavily there, you can visibly see the CSO happening. That's the pollution you see. What you don't see, of course, are all the chemicals, right, that are overflowing from this, that normally would go through the sewage treatment process and are now ending up in the water, right? So it's this chemical pollution that's our primary concern. Of course, right, this is San Francisco, right? Not many people actually dump stuff straight in the storm drains anymore, but you zoom out, right, and nobody's dumping anything, but this is, you know, you live in a city, and there's just all kinds of stuff that piles up, right? So, and of course, you got rainwater running over this, so it's no wonder, right, that we have these low concentrations of, you know, thousands of chemicals that are going out into our nearshore areas. Okay, so the chemicals I'm going to talk about today do not have an accessible name. Polycyclic aromatic hydrocarbons. They're called polycyclic because they have multiple, these are their chemical structures here. One of these is what's called a benzene ring. And they have multiple benzene rings stuck together in various configurations. This is, in fact, this was kind of pioneered by the Ock Bay lab in Juneau. This graph is an output of the kind of analyses that they did. And if you go from left to right, the size of the compound goes up. So 2-ring compounds are purple here, like this naphthalene. 3-ring compounds are these guys here. 4-ring compounds are here. And here's a 5-ring compound, but notice that you don't really see any of the 5-ring compounds in there. These are present in the oil, but they're very, very low concentrations. These things are actually created by combustion. So when you take a fossil fuel and burn it, you tend to create these higher molecular weight compounds. And if you go and look at sediments in Puget Sound, then you actually start to see high concentrations of these things. These things cause cancer in fish, cause cancer in people. This is also the same thing that might give, if you eat too much barbecue and you get gastrointestinal cancer, that it's because of this. So these are the chemicals that we're talking about, and this is in water, right? So if you spill oil on a beach, and then you've got water flowing across that beach, this gets dissolved out of the oil, and it shows up. So this is an oil spill pattern, okay? And this is from this is actually Alaska North Slope crude oil that generated this. If you look at an urban stream, this is Longfellow Creek in West Seattle. You can see there's an awful lot of overlap. Right? And if you look back at that cell phone video I showed you, right, where we got all this slow trickle of everything that's coming from both tailpipes and leaking oil pans and all that kind of stuff. Now you start to, remember I said about the red compounds, so you start to see the red compounds showing up and these yellow ones because of combustion, right? So it's a kind of overlapping pattern of an oil spill plus this combustion stuff. But so from a biological modeling standpoint, the oil spill is actually simpler. So that's one of the reasons we study oil spills very carefully, because just to understand what these compounds doing, oil spill is like pH is in isolation. Stormwater runoff gets more complicated because you have this pyrogenic, the burning signature. Then on top of that, you got all the other stuff from stormwater, right? You have all the pharmaceuticals and everything that are, or pesticides coming from lawns, et cetera. So in order to understand the really complex mess that stormwater is, we're first trying to understand the sort of simple mess that an oil spill is. Okay, and this was other pioneering. I'm surprised, were you around? Yeah, there he is. And I didn't even know Adam was going to be here. That's why it's great to be here. So this is pioneering work, right? So in the early days, when the Alaska pipeline was first set up, this is what these guys were tasked to do. So Jeep Rice, who led this group, they went and collected a whole bunch of Alaskan fish and shrimp and invertebrates and worms, because when the pipeline started and they started taking the oil through Prince William Sound from the Valdez terminus, they wanted to understand what would happen if there was an oil spill. So this was traditional toxicology. So they did what's called an LC50 test, basically, or the lethal concentration that kills half the animals. But the animals they took were all mainly bigger ones, right? They took, juvenile salmon and juvenile, well, Adam, you could probably rattle off all the different species. But it basically took relatively high concentrations to kill the animals. And they found that pelagic fish were the most sensitive. But the concentrations that killed them, when they measured aromatic compounds, it was in the parts per million range, or milligrams per liter, okay? Parts per million, that still sounds pretty low, but from a toxicology standpoint, that's actually pretty high. And why we knew it was high was because when the Exxon Valdez spill actually happened, Jeff Short, the chemist in that group, went out and did water sampling. He took water right under the oil slick, when they all thought it was going to be this really high concentration. And when they measured it, the aromatics came out in parts per billion, 1,000 times lower than what they knew would kill pelagic fish. So, of course, Exxon is like, well, it's no big deal, right? It's 1,000 times lower than, than what kills animals. Okay, so we're in Bellingham, right? Home of Boundary Bay and Chuckanut Brewery, right? Big people can drink a lot of beer, right? The reason we can drink a lot of beer is because we have an enzyme called alcohol dehydrogenase, which metabolizes it, right? Okay, well, big fish can metabolize PAHs. They have an enzyme called cytochrome P451A that metabolizes. Just like people can take a lot of alcohol and it doesn't kill them, fish can take a lot of oil and it doesn't kill them, okay? But what about alcohol in human embryos and fetuses, right? We all know that that is a bad thing, and that's because the smaller animals don't have the metabolic capacity. So what happened in 1989 is we had a surface slick that was whipped up into what's called a mousse by the storms that came through in the springtime, washed it onto the shoreline. So we had this big oil bathtub ring, okay? And it coincided with the spawning of herring and the spawning of pink salmon. So this is herring. Well, you guys, I mean, you're out there. Maybe some of you have been lucky enough to see these whitewater events, right? When the herring nearshore spawners come in and deposit their eggs on eelgrass and other nearshore vegetation. So this is in British Columbia, but this is a huge whitewater event, right? This is all herring, milt, and eggs mixed together, right? So they come in and spawn in these massive numbers and deposit sticky eggs on eelgrass and macroalgae right next to the shoreline. Pink salmon in Prince William Sound, pink salmon generally are kind of lazy, right? They don't migrate really far up into streams and rivers like Chinook or sockeye do. They kind of flop up on the beach and they'll build their redds and deposit their eggs in the intertidal deltas, right where the stream is crossing the beach. So you have an oiled beach, you have herring eggs right next to the oiled beach, and you had salmon eggs deposited in streams right next to the oiled beach. So again, there was already some information indicating that young developing fish would be more sensitive to lower concentrations of these compounds. And so it was the field sampling that was done after that oil spill when they started and they identified malformed larvae and pumped redds, right? They sampled redds in here and they found a lot of dead eggs and things like that. Okay, so this gets us into fish development, right? So development of all vertebrates, right? You start with, the egg is a single cell, right? You have a single cell egg, it starts to cleave, right? And you get the body axis, you get the formation of the primitive organism on it, and in a fish embryo, this is actually herring, right? So this is Atlantic herring development, but Pacific herring is virtually identical. So inside the egg, right, you have the embryo curled up in there, or all these stages, and right here, this guy right here, sorry, I'll try to go over here, right, this is a hatching stage herring, okay? So these stages are all inside the egg, then it hatches, and then these are the hatching stages all the way up to a juvenile fish, right? So this is a fish this guy's a forage fish now, right? This is a 3 centimeter fish. This is when you start to see the schools, and this is what all the birds and everybody are liking to eat, okay? But these stages back here, this is the fish, this guy can tolerate relatively high concentrations of stuff. These guys can't because they don't have any organs yet, right? All the organs, the liver is the main metabolic organ of all of us, right? The liver is what protects you from alcohol. The liver is what protects fish from PAHs. If you look at flatfish in Puget Sound living on pH-contaminated sediment, what do? They get liver cancer, right? Because the liver is the organ that's doing all the work to metabolize this stuff. Well, there's no liver yet right here. So that's the reason these guys are sensitive. The other thing is that these the fish eggs, they're filled with yolk, they're lipid rich, and these pH compounds are hydrophobic or fat-seeking. They're lipophilic because they're just carbon and hydrogen. So oil and water doesn't mix, right? But they have enough water solubility that they'll move from the oil into the water, and then as soon as one of these compounds, if these guys are bathed in them, those compounds would rather be inside the fish egg than they would in the water. So fish eggs are very effective at bioconcentrating these chemicals in the water. So you can have really low parts per billion in water. They'll accumulate to 100 or 1,000 times higher inside a fish egg because of this process of them being fat-seeking. Okay, what this comes down to is this right here, right? So this is the exposure differences between the embryonic and juvenile fish and why oil doesn't kill the juvenile fish or adult fish. And you have a developing fish embryo, and these fat-seeking compounds will just go right across the eggshell and accumulate inside the embryo, right? The pathway is direct absorption right across the skin. As I said, the organs are just developing, so they don't really have a metabolic protection. because they don't have a liver yet. So that's why you get this bioconcentration, and that's why they're so sensitive. You know, whereas an adult fish, its skin is protected by scales, right? So the compounds don't go right across the skin. If these animals are going to take up chemicals, they're going to take it up over the gills, right? Because that's what's in direct contact with the water, or they'll ingest it and they'll take it up through the gastrointestinal tract. Well, an adult fish, if it takes it through the gastrointestinal tract, it'll go through the liver first. That's what's called first-pass metabolism, and that's what, same thing with alcohol, right? That's what protects us from killing ourselves with alcohol unless you overwhelm it. That first-pass metabolism really protects these larger fish. So does that make sense for everybody? Okay, so here's the other thing about embryos, and fish embryos in particular, and this is one of the reasons that biologists in general like to study fish embryos, because they're way easier to study than mammalian embryos, human embryos, because you can't look at them directly. These guys are developing outside their mother. Not only that, this is an herring embryo. They're just spectacularly translucent. So you can just put them on a really simple microscope and literally just look at it. And the animal tells you what's wrong with it. And that's why this guy, Victor Hamburger, who is a very famous developmental biologist working primarily with chick embryos, right? He said, our real teacher has been and still is the embryo, right? And the embryo is always right. And that's the beautiful thing about this, is with microscopes, you can just take pictures and take videos of these guys, and you can't argue with it. That's one of the things from a Dange assessment standpoint. That's one of the things where it's easy. You can put this picture in front of a judge, right, and say, "Do you see that?" And they're like, "Yeah, I see that." So the embryo does not hide anything. This is a herring larvae. This is a digital video of a hatched herring larva. This is the heart, right? So these things are just it's beautiful. You just put them on there and look in the microscope, and you can watch the beating heart. And that's really what allowed us to. The story that I'm about to tell you is entirely dependent on the fact that these animals will just lay down and show themselves to us, and we just had to look at them in the microscope. Now, there is a lot of kind of fancy, less accessible molecular biology and stuff like that we're doing these, but almost all this is based simply by looking at the animals. This is an Atlantic haddock larva, hatching stage larva. Here's the heart beating. Here's the yolk. Actually, this, where is it? Right here. This little ball is the liver. Okay, that's the liver bud. So it's got it right over here. That's the liver. This is the intestine, the stomach and intestine. This is the brain. I mean, literally the whole animal. You can just look in the microscope and see every single tissue just with the right kind of optics. Okay, so this is what the Ock Bay lab discovered, right? So this is a normal pink salmon fry here. This is one exposed to oil. This is a herring larva, newly hatched herring larva, Pacific herring. This is one exposed to oil. Can you guys see the difference? It's pretty obvious, isn't it? Especially with the salmon, because you look at its yolk sac and you can see that it's bulged out and there's this clear. It's filled with fluid, right? There's this clear space. It's a little bit harder to see on the herring. Now, the first thing you see is the tail's kind of squirrely, but look at the yolk sac, right? And you see the difference here, and you see all this clear space in the yolk sac because what's happened is they've got extra fluid in them. And this is heart failure. Okay, and it's the same thing as heart failure in humans, right? In humans with a bad heart, it's called congestive heart failure because your pump, right? The heart is a working pump. When it's not pumping properly, the fluid backs up. If you're not perfusing your kidneys and your liver the same way, fluid accumulates in the abdomen. You get fluid accumulation down in the ankles, pedal edema. Fluid accumulation in the lungs. That's the whole reason it's called congestive heart failure. Those fish that I showed you on the previous slide, in essence, have congestive heart failure. Okay, the other thing is that what we call, this is all kind of a little bit arbitrary. When we start talking about, remember, we're talking about parts per billion concentrations. So when we say high, we're talking about high parts per billion. You go to parts per million, and that's like way high, or way, way high. So at these relatively high concentrations, this is now, these are Arctic cod larvae going from a control to increasing concentrations of oil. These numbers are actually total oil, okay, not PAHs. The PAHs tend to be like 1% or less of the total chemicals that are in oil. This is a herring larvae that was exposed to 4.2 parts per billion. And those were the kind of concentrations that were right under the slick after Exxon Valdez. 10 parts per billion, 4 parts per billion, those were about the highest that they saw in Prince William Sound. Deepwater Horizon, most of the water samples even were coming out in those, tens to the highest ones were maybe 100 parts per billion. At these increasing concentrations, right, you can see how these larvae are really messed up. Their jaw is messed up, and part of the reason their jaw is messed up is because the heart is developing right underneath the jaw here, and as the fluid accumulates in the yolk sac and around the heart, that disturbs development of the jaw. Well, this is lethal. This is acutely lethal. This thing, as soon as it absorbs its yolk, it's not going to be able to feed. It's not going to be able to go out and capture the tiny plankton that these larvae feed on. Remember, these larvae these guys are like 6 millimeters long. A herring larva is about 8 millimeters long. So this is a lethal effect, okay? And so what I'm kind of getting at here is in toxicology, we call this, there's a dose response, right? Everything is dose dependent, right? At high concentrations, you get really severe malformations. As you go to lower and lower concentrations, the malformations aren't so obvious. But when we look closely with digital video at the heart, we can still detect changes there, and that's kind of what I'm going to show you. Now here's what happens at the low end, right? So these were critical experiments that the Ocbait lab did, and they could do this with pink salmon because pink salmon are really a great model species because they have this nice, precise 2-year life cycle. They're not like Chinook or coho that may, Chinook may be 4, 5, 6, 7 years old when it spawns. Where pink salmon, they're always 2 years old, right? So they basically spend the first year of life, the first year is down in the red, making a smolt. Once the smolt goes out of the ocean, it's only in the ocean for 1 year and then it comes back. So these guys using hatchery technology and coated wire tags, they did these amazing basically tag and release experiments where they exposed tens of thousands of pink salmon eggs, hatched them out, exposed them to low concentrations, right? So if you look in the previous slide, it was a concentration that gives you basically an externally normal looking fry. It wasn't a fry that was filled with fluid, it was normal looking. And in the hatchery, then they put coated wire tags in them and released them and waited a year for them to come back. And with increasing concentrations of oil, and these guys did this over a decade, they did it 3 times, and reproducibly got a 40% reduction in survival to fish that were exposed to parts per billion oil. So this kind of raised, for us, this was, the obvious question was, is this related to the heart, right? We knew that at these high concentrations there were these really serious heart defects. What we wanted to know was down at lower concentrations, is it also an effect on the heart that's kind of driving that delayed mortality? It also kind of it points out the whole, how do you quantify losses, right? Because if you looked, the year of the oil spill, and those fish are 2 years old and they're not coming back. The year of the oil spill, you do your damage assessment, you're like, oh, we lost this many eggs, but what about the eggs that were exposed to low concentrations that weren't obviously lost? How do you quantify that, and how do you get recovery for that? Okay, so humans and fish have very similar heart structure, right? We have a 4-chambered heart, right, because we have this dual circulation between the lungs and the rest of the body. So we have an atrium. In mammals, there's 2 atria and 2 ventricles. The ventricle is the bigger, more muscular chamber that sends the blood out to the body. So, right, fish has a single atrium and a single ventricle. This white thing here is the bulbous arteriosus, which is the same thing as basically the aorta, right? It's the outflow tract. This thing right here that kind of looks like a tulip bulb. Now this is kind of backing out a little bit, and this is a little bit of the fire hose stuff, but I'm just going to go through this. But basically what this shows is that just by looking in the microscope, we can watch the heart develop from its very beginning, right? And it starts out in the early embryos just 2 patches of cells that are the cells that are going to be the atrium or the ventricle. They fuse and form what's called a cone. You can, in the microscope, it literally looks like a cone. This is a belly view and this is a side view. And then it turns into what's called a heart tube, and this is just a very simple tube with a thin wall, this is when it actually starts to beat. Okay, so in a fish embryo, in the microscope, you can, in real time, this is actually a red drum larva from the Gulf of Mexico, which those guys are living at like 28 Celsius. They develop in 24 hours. So literally, you can sit in the waterscope and just watch the heart develop. You watch the first contractions. You watch it start beating. You watch it form, until it goes through this. It goes from this linear tube through a looping, and then the ventricle starts to grow and turn into this big muscular thing. The main point of this is, as I said, it begins to pump right here. It's the first organ to actually start working in the developing embryo. And it's working while it's being built. So kind of like the proverbial, trying to work on an engine while it's running. If you mess up its function, you mess up its form. So form and what happened there? I went too fast. There we go, right? So form and function are so intertwined in the developing heart that you can't disturb one without disturbing the other. And if you disturb, and they feed back on each other. So if you disturb the function early on, you change the shape early on. If you change the shape later, the function is going to be different. So it just keeps going back and forth. So that is the explanation for why putting a chemical in here that's going to disturb the function of the heart, of the developing heart, is really going to mess it up and lead to heart failure. I don't know why it wants to skip through that. Okay, here's another thing about fish hearts. Can you see the fish? Yeah, so that's a flatfish, right? So these guys, flatfish, sculpins, the fish that just like to hide out and ambush, they kind of have a flabby saccular heart, right? These guys don't swim for long periods. The fish like herring, you don't really think of herring as like this super strong fish, but they really are because they swim constantly. They never stop. They're these planktivorous fish, just swim, just keep swimming, right? Like, that's my favorite movie, as you can guess, but... So salmon, right? Salmon are fish that migrate very long distances. Tunas, right? Tuna is the Maserati of the sea, the apex predators, right? These fish that do really high-speed, muscular, continuous swimming, they have a pyramidal ventricle, and they actually have a coronary circulation, just like we do. The fish that are ambush predators, they don't have a coronary circulation. So this heart structure for these fast, continuous swimming fish is tied to a high cardiac output, right, which supports that athletic ability. And we know this, right? Babies that have congenital heart defects don't grow well. I don't know if they're pretty common. Some of you guys, I don't know if anybody here was born with a congenital heart defect or had a child, but that's kind of the bread and butter at Children's Hospital. And we can take care of kids that have heart defects, and they can lead a normal life. But when you look at how they grow, so the white dots here, the clear dots, those are infants that don't have a heart defect. The black ones are all the have heart defects. And if you look at the growth trajectories, right, so this is how old they are after being born and their weight, right, you can see the babies without heart defects are growing like crazy, right? But look, the guys that have heart defects, a lot of them are flat. They're not growing. So what is it about having an impaired heart that leads you to not grow well? Well, there's a lot of studies. It's kind of a no-brainer, right? You're putting a lot more energy just to keeping your heart going and just to keeping your basic metabolism going. The food that you consume goes into just your basal metabolism instead of going into growth. So this concept is exactly the same. If you impair the heart of a larval fish, how is it going to grow? How is it, and the thing about fish is there's a huge literature on size-selective mortality. Little fish get eaten more than big fish. It's kind of a no-brainer. These are larval sardines, and this is a complicated graph, but basically what this line shows is that if you're a big fish, you pretty much stay in the population and you survive. If you're a little fish, you disappear. And this is for sardines. There's again, with hatchery operations, right? The salmon hatcheries that we have here, the importance of those, I know there's a big debate about what they contribute to wild fish, but what they have allowed us to do is to really make correlations because they tag these fish, they know their size at release, and they know when they come back. So there's a massive amount of information on how big you need to be as a juvenile salmon in order to survive in the wild. In Prince William Sound, they're doing tracking the herring populations there. That's one of the primary things they're doing is trying to figure out how big does a baby fish need to be in its first year of life if it's going to recruit and join the population as a reproductive adult. Okay. All right, so now we're going to get to how exactly does oil cause heart malformation. I kind of told you that already because you have this working, this heart that's developing, that's working while it's developing. Well, I'm only going to talk about, this is a 30-minute talk by itself, but the heart, of course, is a pump that's driven by the electrical, what we call excitable cells, right? Muscle cells and the pacemakers in there are excitable cells, and it's actual electrical activity of those cells that drives them. And you see that in an electrocardiogram, right? That's what the lines on the electrocardiogram show, are the electrical activity measured from surface electrodes that you put on the outside of the body. Well, the electrocardiogram in a fish looks exactly the same as in humans. And you have what's called a P wave, which is basically the pacemaker. That's the electrical activity of the pacemaker. The QRS complex is the contraction of the ventricle. And then the T wave is basically the resetting of the whole cycle. And one of the main side effects of human drugs is actually blocking this ability to reset, and it prolongs action potentials, and it prolongs the duration of this QT interval. Does anybody remember Seldane? Seldane was the first non-drowsy antihistamine. It was taken off the market because it caused some people, their hearts to have serious arrhythmias and stop and basically stop and have sudden death. That's one of the main reasons drugs get taken off the market. In all pharmaceuticals, one of the first line of screening is for this activity, for blocking this ability to reset the heartbeat each time. Well, the oil compounds actually do that exact same thing. So they mess up the electrical activity of the heart. If you're an adult, if you have a fully formed heart, as I just alluded to with Seldane, if you cause a serious arrhythmia, the heart might stop and you die. In a developing fish, it's not going to kill it. But it's going to cause a malformation because of this form-function cyclical relationship. So that's exactly what's happening. And you can see this, again, in digital video. So these are embryo stage. So these guys were cracked out of their eggshell. These are embryo stage herring embryos. And this is a normal heartbeat. And then these are guys that are exposed to increasing concentrations of oil. I don't think all the movies are running. I may need one. Yeah, now they're running. So here you have a nice regular heartbeat that's 104 beats per minute if you sit there and count it. This guy, the heartbeat is regular, but it's going about half the rate, 54 beats per minute. And if you watch this one, it's like, I'm going to beat. It's going really slow. I'm going to take a rest. I'm going to beat. At some point it makes a long, where's it going? Yeah, there, I'm going to rest a little bit. So that is a very irregular rhythm. That rhythm, if this rhythm were happening in one of us, we'd feel really sick. And we'd be in the hospital. We'd be electroconverted, right? Fish embryos are so tiny that they actually get oxygen by diffusion. And notice you don't even see any blood, right? These herring embryos don't even have any blood yet. Herring larvae are so small and living in this cold, oxygen-saturated water, they don't even have red blood cells until they're probably 6 weeks old, because they're just sucking up oxygen from the water. That's why you can knock their heart out and it actually doesn't kill them. So they can live with a heart malformation for quite a long time. But they're not going to, well, I'm going to get to that. They're not going to. All right, so that kind of gets back to this. Okay, so this is, these are herring larvae again. Now, if we go down to these lower concentrations, and this is kind of what I'm getting to about the, not these high concentrations that cause these really serious malformations, but these are taken from digital video. So you saw the heart beating earlier. And if you look here, this is lower magnification, this is higher magnification. V is the ventricle. So this thing that kind of looks like a little windsock coming back here, right? You guys see the windsock right here? That's the ventricle. This is the atrium. This guy was exposed to oil. And here you can see there's fluid accumulation, right here. But look at the ventricle, right? The ventricle hasn't formed this windsock that comes out, so it's small. And that's something that we can actually quantify by getting, what we do is we line up, we'll take 100 of these pictures, and we can go in and trace the size of the thing, and we actually put a number on this. and that's shown here. So again, look at the concentrations. The highest is 4 parts per billion. This is our control. This is basically detection limits, in the control water. This is fluid accumulation. So this is scoring animals that have congestive heart failure. But notice that only, at this concentration, only 30% of the animals have what you could say is visible congestive heart failure. 70% of them look normal. But if you look at the size of that ventricle, you can see that we get this dose-dependent increase. And even at 1.3 parts per billion, even though there's none of these guys have congestive heart failure by the measure of edema, here at the lowest concentration, they have a significant reduction in the size of their ventricle. So their heart is starting out a little bit smaller. And you can see that here. So this is traced out again, right? So the ventricle in a control animal is traced here. Oh, this is now after a week, right? So the slide I showed you before was at hatch. If you grow them for a week, the hearts start to grow, but they're still smaller. And the experiments that we're doing right now is trying to track that, right? Trying to track, the question is, do they catch up at some point? Actually, they do catch up, and they actually overshoot. These are pink salmon hearts. So we've done these same experiments in pink salmon where we exposed, this is basically replicating the studies that Ron Heinz did with Auk Bay, the tag and release studies. So we're using concentrations that are the same. This is now in these externally normal fish. I thought I had, I guess I did. I have a picture of pink salmon coming up. But if you look at the shape, these are juvenile fish. So these are fish now that were exposed as embryos, but now they're 7 to 9 months old, growing in a hatchery. And if you look at the shape of their ventricle now, you can see that the exposed one, it's a little bit longer, and that's shown in the length to width ratio. So again, just in a bunch of pictures, if you measure the width of the ventricle and the length of the ventricle and divide those to get a length to width ratio and plot those on a graph, that's what you get here, right? So we get an increasing length to width ratio that's dose dependent. So the more oil the fish was exposed to, its ventricle starts to elongate. We think actually started out smaller, right? In the larva, it was smaller. 7 months later, it's now bigger. Now, if any of you guys know, I don't know, does anybody have heart? Has anybody experienced had a family member with heart failure? What happens when a failing heart gets bigger, right? Hypertrophy. What happens when a heart starts to not function well? It starts to compensate by the process of hypertrophy, where it tries to grow bigger. And when we section these hearts, and look, these hearts actually have signs of hypertrophy. So these fish have a weakened heart, even though it kind of caught up. Now it's overcompensating, which is a sign that it's not functioning as well. Well, how do we actually measure heart function in fish? Same way we do in humans. We put them on a treadmill. So we give them a stress test. This is a move. No, maybe that movie's not working. Shoot. So this is a lap pool, or a little treadmill lap pool, whatever you want to call it. We actually have a juvenile herring in here. So there's a little motor in here, right, that's generating a stream, just like a lap pool that the fish swims against. And you swim those fish basically till they're out of breath. And the speed that they swim, we call critical swimming speed. It's a duration test. You can actually quantify how athletic is the fish, how long can it swim. And this is a pink salmon. This is like, Ray Troll's got to have something on this, because they're not pink, right? They're blue. Why do they call them pink salmon? No, there's all these. Well, forage fish the same way. These forage. I mean, a juvenile pink salmon basically is a forage fish, right? And they got that beautiful blue color, so the birds can't see them from the top. And they're just. But this guy's oil exposed. But he's this beautiful little fish that looks totally normal. But you put him in the treadmill. These are the controls. This is body lengths per second, right? So we convert centimeters per second to how many body lengths can the fish move through the water. Well, a normal pink salmon can swim about 6 body lengths per second. If they were exposed to oil as embryos, they lose a body length per second. They're down at 5-ish. And that's a pretty significant reduction in swimming speed. This is juvenile herring. Same thing with juvenile herring. Controls were swimming, this is centimeters per second. This is converted body lengths. So a 7-month-old herring can swim about 5 body lengths per second. If it was exposed to oil as an embryo, it can only swim 3.5 body lengths per second. Okay. Okay, so this gets to what's the no effects concentration. Actually, if I go back here, so remember we talked about parts per billion. These guys were exposed to 0.23 parts per billion, 230 parts per trillion. And this data is published. Our unpublished data is this. So we just did, actually these are, I know people always ask, are there differences in stocks? We've done this with fish from San Francisco Bay. We've done this from fish with Yaquina Bay down in Newport. We've done it with all the Puget Sound fish. This is Cherry Point fish right here. We just did this exposure last spring with Cherry Point fish. This is the ventricle shape at hatch on May 5th. 110 parts per trillion pHs. This is the control. Notice that these two middle, this was oil gravel columns, the middle doses are below detection limits. Now we did not have an effect on the shape of the heart, but here at 110 parts per trillion we did. But this is the intriguing thing. So this is measuring that. Remember I told you the enzyme. The enzyme cytochrome P451A is the protective, even though baby fish don't have a liver, right, they will induce this enzyme in response to oil exposure. And we're getting significant CYP induction at concentrations that our chemists can't even measure, okay? So the embryos are actually better at detecting this stuff than even our chemists are with their $100,000 instrument, right? This is the length of the larvae at hatch. Remember, these two concentrations here were below detection limits. So even though there wasn't an effect on heart shape, there's an effect on total length. This is now just last week, okay? This is looking at that length width. So the fish are now, how many months is that from May to October? 5 months old, right? So the fish are like 5 months old. Here at 110 parts per trillion, right, we have an increased length to width ratio, just like we saw in the fish that were exposed to slightly higher concentrations. So where is the no effects concentration for herring? You know, it's somewhere near the detection limits for these compounds. These concentrations down here are literally at background levels in urbanized Puget Sound. This is a complicated slide. The main thing is this is with cod, right? There's a huge amount of studies on cod. So cold water fish, there's a massive amount of information saying that the first year of life and surviving the first winter are critical, and that's what determines whether or not fish are going to make it into the reproductive population. And it's related to how big they get for the first winter. If you think for herring, it kind of makes herring in Prince William Sound, it's very informative because in Prince William Sound they've got ice floes all winter, and juvenile, year zero herring hide under the ice. If they're not big enough and if they're not fat enough to survive the winter, what do? They go out and try to find food, right? They forage. They leave their protective ice thing to go to find food. If a herring goes to find food, what happens? Somebody else finds it as food, right? That's the whole size-selective mortality thing. So this is all converging, right, with a lot of fisheries data saying that, for these fish that live in these cold climates, that first year of life is critical, and how big they get before the first winter is really what determines when they survive, right? So here we have this, sorry, I forgot I put this in there. The other thing we're doing is right, I just said the no effects concentrations is probably at background levels in Puget Sound. And I showed you how the urban creek looks exactly like an oil spill. So what we're starting to do now is actually take storm highway runoff from down at the Mountain Lake Lab. We've got State Route 520 goes right over our parking lot. We got a storm drain that drains right in our parking lot. So we just put a big tank under there and collect it. WashDOT was a little bit annoyed with us at first, but we're trying to help them figure out how to clean the stuff up, right? So that it doesn't do any damage. But we exposed herring to stormwater, during this critical developmental period. And what are we getting? We're getting the exact same. I'm going to skip through this one, right? But here, look, here's your control herring larva. Here's the one exposed to stormwater. Guess what? It's got a heart problem, it's got a jaw defect, and it's a little bit smaller, just like you get from an oil spill. And here's the effect on the heart. This is the graph showing the size, again, that size of that ventricle as it grows. So the stormwater runoff, even though it's way more complicated, it's got all this other stuff in it, but it also has an oil spill, and it's giving us the exact same effect as an oil spill down at these really low concentrations. The concentrations in 520 runoff are 10 to 13 parts per billion, which that's under the slick from Exxon Valdez. So what we've done is we kind of have this, we're trying to have this cause and effect chain, right? So we have these compounds, pH compounds in oil that are toxic to the heart. They cause an abnormal rhythm. And during development, right, because of the form-function relationship, causes a change in heart shape. Because heart shape is so critical to cardiac output, that affects the cardiac output of the fish, which in turn reduces its athletic ability, its aerobic capacity, its capacity to swim. But the other thing is that we think and there's actually, I haven't talked about it, there's other indirect ways that we think that this effect on the heart is basically altering bioenergetics and growth, just like it does in babies with, in pediatrics we call it failure to thrive. Have you guys ever heard of that? I mean, when you have an infant that's not growing and you're trying to, that's how a lot of congenital heart defects are discovered actually, because you have an infant that's not growing. So that's what we think is happening with these baby fish, but with, unlike humans who can go to children's hospital and have somebody take care of them, right? We don't have children's hospital for fish. Those guys are out there on their own. So again, it kind of gets back to this, right? So even though this is all about oil spills, the herring, right? We're literally just looking at the fish, and the fish are telling us that this stuff coming off the pavements at these concentrations that all of us are contributing to are, in all likelihood, having an impact on them. And that's it.
Whether the emulsified oils and whether or not they have a more significant adverse effect on fish, and then address that as far as oil dispersants as a way to control oil spills.
Well, so that's what I was going to add. When you say emulsified, is that you mean when using dispersants to
Well, there's other ways that oils are emulsified, such as toppings on blacktop and things like that.
Yeah.
Specifically for oil spill dispersants.
Well, so the thing is so oil spill dispersants are basically detergents, right? That break this whole like fat-seeking thing, right? Detergents will, coat the outsides of the oil droplets and make them smaller. In fact, the whole point of dispersants, and that's the reason they used them in the Gulf of Mexico. And in the Gulf of Mexico, you can make an argument that it actually worked because it's such a hot environment. You disperse the oil into smaller and smaller droplets. That makes it more bioavailable, makes it easier to biodegrade, right? To have bacteria break it down. Well, again, that's fine if you're a big animal. But what makes it bioavailable to bacteria also makes it bioavailable to fish eggs. So the whole dispersant thing, that at least because we do work with the oil spill response people, it's the trade-off. So you have to what we tell people is which animals do you want to kill? You want to kill the seabirds and the marine mammals, then leave the stuff on the surface. You want to kill the fish, then disperse it and put it into the water column. But so that's why understanding the ecosystem is so important and knowing what animals are where at what time. And you can kind of argue if you have an oil spill and there's no spawning fish around, then you're like, yeah, drive it into the water. Gulf of Mexico, where you have this warm system where the thing about these cold water environments is biodegradation doesn't happen nearly as fast. And a lot of it in your head, you think, oh, these cold water animals, everything is so slowed down. You'd think that the uptake would be slowed down. You'd think that it's the flip side, is that we find the cold water animals are actually way more sensitive. And it probably has in part to do with the metabolism, but in part it has to do with, this is one of the things we learned from Deepwater Horizon is tunas. So Bluefin Tuna, which, right, they dive super deep. I mean, you go down 1,000 feet in the Gulf of Mexico, what's the water temperature? It's 4 degrees, right? It's, up at the top, it's 80, 89 degrees, and, 1,000 feet down, it's 4 degrees. Well, these tunas, right, they can dive that and come back. So their heart physiologically is adapted for cold temperatures. And the adaptations for cold water are what create the sensitivity. So that's why our herring and Arctic cod and things like that are sensitive. And if you look at the fish in the Gulf of Mexico, the most sensitive fish were the tunas. Fish like red drum, which are kind of these lazy big goldfish that hang out in the shallow, they're not as sensitive. And it's because this gets into the really detailed, physiological differences between fish that have broad temperature ranges versus short, narrow, warm temperature ranges. So kind of a long-winded question. So the whole dispersant thing is all about trade-offs and understanding what animals are where at what time.
So I was in the middle of Harvey a couple of months ago.
Oh, I'm glad you're here.
Thank you. Flew in the night that hit Corpus Christi and Rockport. Spent 2.5 weeks down there, and probably one of the most grossly underreported things about that whole hurricane was what I consider to be the largest single storm runoff disaster in the history of this country, because there was stuff that was flowing out of all the refineries along the coast, out of Houston itself and the surrounding county and so on and so forth. So I guess it's kind of a suggestion and a question. Have you heard anything from your peers down in an equivalent agency down the Gulf Coast about what they're going to do in the way of the same kind of studies that you've been doing, and you've alluded that they have done also, because with the amount of toxins that flew out in Galveston Bay has been flew out for decades already, despite what's going on. Is that kind of information going to be, I hope, helpful?
It's a good, interesting question. The whole time, we were waiting to get the call, right? We went down for Katrina, right? The group I'm in, so when Katrina did the same thing, right? They had this huge toxic flood from Lake Pontchartrain and everything out in the Gulf of Mexico, and they closed the fisheries, right? Shrimp fisheries were closed. So we were down there monitoring after Katrina. So after Harvey, we were, like I said, are they going to call us? And we never got the call. All I can say is, having worked, on Deepwater Horizon for 6 years and spent a lot, I'm a Southerner myself. I grew up, I'm from the Florida Panhandle. All I can say is Texas is different. That's all I'm going to say. Texas is different. Pacific Northwest is different.
Thank you. John, are you going to be, I think we're out of time for...
Yeah, I'm sticking around.
But,
Yep.